Today's April 11th, 2020. I'm Brent Nally, and I'm really excited to be partnering with Leaf and Lifespan.io for a new video series where I will be interviewing longevity thought leaders. And I've been doing this for about the last year and a half on my YouTube channel. You can check the link in the description below. And I interviewed Dr. Alex Zhavoronkov, who is our guest for this first series. And we're going to be focusing on COVID-19 today, as you might have guessed, due to the current state of the global pandemic. And there's actually a very interesting and important tie to the research that Leaf and Lifespan.io and Insilico Medicine and Alex are doing to COVID-19 that has to do with biological aging. So I'm really excited to be here today and be here with Alex. There'll be a link in the description below to our first interview, which was in late January 2020 on my YouTube channel. It's just titled Brent Nally. Alex, welcome.
Great pleasure to be on your show. Very much like Lifespan.io. This guy's managed to achieve so much in so little time and build a fan base. And you partnering with Lifespan.io is, I think, the very good decision, especially in this turbulent time. So very happy to be on your show. I am currently being quarantined in Hong Kong. So just arrived in Hong Kong from London. You've probably followed my trip on some of the social media. It was a lot of fun. So right now I've got this bracelet, which basically resembles a prison bracelet, or so it doesn't allow me to leave the apartment. So and I'm very grateful for the new Zoom feature of having a background like that. So also using AI and deep learning. So you can see that there are tangible applications of AI already. And from here, I'm basically taking command of a large number of research troops worldwide. We've got researchers in 6 countries or regions as Insilico. And of course, we continue working full steam ahead. So regardless of the pandemic, because we're virtual, and we are Insilico. So we just we do pretty much everything in the computer, in the cloud, and have a pretty large laboratory base, more than 80 contract research organizations that we can outsource our research to, and a network of about 150 research institutions, including some of the really big names. So many of them continue to work.
Great. And I know that Leaf and the Lifespan.io team and Keith Cometto, the founder and president of Lifespan.io, are very mobile and set up on the internet to be, not as affected as a lot of people, because we're very video conference competent, and I am as well here too. So that's great to know. So before we jump into it, I just want to state the date again. It's April 11th, 2020 today. And the Worldometer stats is what you're going to hear right now. We'll have a link in the description below. So I encourage the audience to check that Worldometer website for the current stats. But as of right now, we currently have 1.8 million infected with COVID-19, about 109,000 deaths, and about 404,000 recovered. So those are the official stats today, and stay tuned. So Alex, we're familiar with your work, from my interview in late January, and I encourage the audience to check that out. You've been known for, your work in drug discovery at Insilico Medicine and your team. How does, what you're doing relate to what you've been doing at Insilico Medicine relate to this period in time? And how did you kind of find out about COVID-19? And how did you kind of immediately realize that you need to nimbly transition your research in this pandemic in these very strange and weird times? So can you walk us through kind of that story and how that's gone about over the last few months?
So sure. Well, first of all, I kind of foresaw that there's going to be some kind of pandemic in the next few years, so to speak, a while ago. So we pretty much tailored Insilico to be able to very quickly discover molecules for diseases that are rapidly occurring. And even before COVID-19 crisis, we were in touch with my really dear collaborator and friend, and one of the people I deeply admire, Alana Spurugudzik, at the University of Toronto Vector Institute, formerly a very famous prominent professor at Harvard, Department of Chemistry. So we are working on anti-infective.ai, so the ability to go after infections really quickly. And we're working on that for about maybe half a year, and we're planning to launch in December, but then we postponed. And right now, if we launch that, it's going to kind of look a little cheesy, right? Because the pandemic is already ongoing.
Right.
But I think a lot of people are focused on developing tools to combat pandemics really quickly and take them head on. And it's just this one stroke too quickly. We did not expect that it's going to come this year. It will eventually come, and I must say that one of the impediments to aging research could be pandemics and economic crisis, because at times when people need to fight for their survival and for immediate economic, basically immediately they need to stay afloat economically and health-wise, long-term plans go away. So people do not think long-term, even we see it nowadays. So my long-term goal is, of course, to set up a big longevity research center on one of the islands that we surveyed recently. Actually, I was there January 1st in China. I think that, Hainan is the optimal place to set up a longevity center, and I've looked at pretty much every place on the planet. So it could be either Chengdu or Hainan. Has to be in China. You need to have substantial patient flow and great expertise and substantial government investment. So, but Hainan is, of course, better for pandemics, right? Because it's an island. So I was in the Boao area discussing this, and suddenly, some of those predictions that I was making came to be prophetic. So January 6th so January 9th to January 12th, that was the time of JP Morgan conference, JP Morgan Healthcare Conference in San Francisco. So those are not the exact dates of the conference, but that's where people convene in San Francisco every year. It's the largest healthcare conference. And during that time, we already saw that there is an outbreak of SARS. So China was already reporting the data. And so those were just light rumors. And strangely enough, again, the largest healthcare conference in the world, nobody was seriously considering it as a threat. So January 15th to January 20th, we started thinking that, it's a great opportunity to test our algorithms, but we also thought that it's going to go away, right? Nobody thought it's going to be a serious disease. So right now, people blaming China, they should not even think about doing that, because scientists in that country predicted this outbreak, 3 years ago. I can share the screen and show you the papers in Nature, where they demonstrate that it is possible for the virus to jump from bats to humans. And at that time, nobody considered it seriously. Everybody thought about, that this is going to be a very temporary outbreak, even less important than the original SARS, 2002 to 2004. And around January 25th, I started approaching my board members to, so that JP Morgan was already over. We resolved all the important things. And I started approaching them for a permission to go after, at that time, it wasn't called COVID-19. It was called 2009 NCOV. And at that time, some of the investors that we have, they were pretty conservative. They said that, what, Alex, is going to go away. If you look at the experience from 2002 to 2004, everybody who rushed into this topic lost, because once the outbreak is over, in a year or two, that's it. So if you didn't deliver a molecule very, very quickly to the market, to the patients, you won't be able to even conduct clinical trials. So it's going to be a dead end. But two of the investors, and specifically, I got great support from Chiming, one of our most visionary and great investors, one of the best investors in the world. The partner at that fund, she said that, Alex, we should go for it, right? Because it's a great way to help the community if it works, right? And if we see this as an extended event, you are going to help people. But in the short run, it doesn't cost you too much to go after it, so why not? And that was around January 28th. We decided to go after several viral proteins, and specifically starting from C3-like protease. It's a protein which controls viral replication. So we quickly analyzed target space, so decided to go after this one, and took us about a couple days to prepare the data. At that time, the crystal structure wasn't publicly available. So we had to do homology modeling on the protein. So basically take the protein sequence, fold it properly to get some kind of representation of the structure. And in that particular, I can actually share my screen and show you what we did.
Yeah, please do. Go ahead and share your screen.
Yeah, so that's, a picture is worth 1,000 words. And sometimes, a picture of the person is worth 1,000 genes, as we say. So I'm sharing the screen. So on this slide, you can see our current pipeline. So we can do target identification and trends analysis. We have generative biology platform where we use generative adversarial networks to synthesize human data. But what we're famous for is the generative chemistry pipeline, where we generate novel molecular structures using AI, and we have the clinical trials outcomes prediction platform, which predicts phase 1 to phase 2 transitions, phase 2 to phase 3 transitions. So we rely on this platform to generate novel molecular entities for a large number of proteins. And let me very quickly switch to another presentation that was, so basically what we do at Insilico is, just to remind you, we have generative biology to identify novel targets. That's not what we're doing here. We do it for all kinds of diseases, also age-related diseases primarily, generative chemistry pipeline, and we have a validation pipeline. Expand my screen so you can see it. So this is how this pipeline looks like in a little bit greater detail. We try to publish in peer-reviewed journals to explain the various pieces of the pipeline, to show that there is a proof of concept. We don't disclose everything there, but we try to give people a sense of direction and make those elements more credible. So to put you in perspective, this is how drug discovery process looks. So the various stages of drug discovery. So usually you start with target hypothesis that can take you months or decades, usually funded by the government and the universities. So thought leaders in multiple diseases are essentially going after the hypothesis behind the disease. Then once you identify a target, so this can take billions of dollars, or it can take minutes. But the thing is, you validate this hypothesis only in phase 2 clinical trials in humans, so many years later. So this feedback loop is very long. That's why the pharmaceutical companies do not really trust the target hypothesis until they have human relevant data, human data. So after you identify a target, so the protein which, or some kind of process that drives the disease, you look for a small molecule that is likely to hit the target, to disable the target, disable the driver. Then you optimize, do preclinical work on mice and other animals. And then you start human clinical trials. Phase 1 is safety, phase 2 is efficacy, phase 3 is both safety and efficacy. And here you usually have, in the traditional paradigm, the years it takes to complete every one of those kind of steps. And the 2010 costs So those are the costs in millions for every step in traditional pharma process. So it's very important to understand that. So in 2010, the drug discovery process, drug development process costed you about, $1.3 billion. In 2013, $2.6 billion. And now it's even more. It's getting more expensive because all the kind of low-hanging fruits are picked up. So we are trying to develop a system so that pipeline that we've developed can address pretty much every piece of drug discovery and try to compress the time it takes to discover the molecules and also increase the probability of phase 1, phase 2, phase 3 clinical trials, and also forecast those probabilities to compress the entire pharmaceutical R&D process. Of course, we won't be able to, in a magical way, do it in minutes, but some of those steps, they could be done in days. Unfortunately, you still need to validate and you still need to do the laboratory work, and you cannot get around a lot of the laboratory studies, and there are many things that can go wrong with those wet lab experiments and mouse and human experiments. So AI will accelerate many pharmaceutical R&D processes, but it's not going to result in ultra magic. So what we did in this exercise, we decided to employ only our generative pipeline and the clinical trials outcomes prediction pipeline. In late January, we decided to prepare the data, so to do the homology modeling, as I mentioned. So the sequence of this C3-like protease was known. So we modeled the protein, fed it into the system. Also, at that time, the crystal structure became available pretty much at the same time when we completed started and completed the homology modeling for 3C-like protease. So scientists in Shanghai, again, I deeply admire, the Chinese scientists who managed to so quickly respond to this outbreak. So a lab at Shanghai Tech, Dr. Rao's lab, now they just recently, so 2 days ago, they published in Nature this particular exercise. So they published crystals, 6LU7 crystal. So we used the crystal as well. And they also had a co-crystallized compound. So some molecule that actually binds the protein weakly, and it's not exactly a drug-like molecule, it's just a fragment of the molecule. So we prepared the data, started the generation exercise. So we basically started looking for novel molecules, imagining those novel molecules using AI that might hit this target, generated about 100,000 structures, narrowed it down using filters to about 100 structures, and submitted them to publication to actually BioArxiv initially. And BioArxiv, those guys were completely in a flux at the time. So they were overloaded with papers. And I think they're a little biased, so I actually don't like that preprint server as much. Because sometimes they just, I don't know, maybe look at the author list and see where they come from, or they look at some other features, or maybe they are scientists also working in this area, and sometimes they just ban you. So this was the first time when we couldn't submit to BioArxiv. They actually rejected it in 4 days. We put it on our website also on February 4th and did a press release, but later we had to submit to ResearchGate, which is a much better platform for preprint servers, because you can also discuss the paper as a community, and it's just a better platform by far. So you see who is commenting, who is interested in the paper, and we got a pretty good community discussion going there. So we submitted later on to ResearchGate and also to ChemArchive, so they accepted. And also quite important to note that this was the Chinese New Year, right? So this outbreak happened during the largest migration of humans in every year, and basically in history, so more and more people travel. So that's when China locks up, right? I mean, China stops their business activities, and that's where a lot of people go home. And at that time, you cannot really synthesize anything there. You cannot manufacture anything there. Everybody's gone, right? Gone home. And at that time, we couldn't really synthesize. So we decided that, okay, we're going to published those molecules and asked for human medicinal chemists to feedback on these molecules, to basically comment on them. They got some really nice feedback. So some community effort was made to comment on those molecules, because some of our previous studies that we published on Nature Biotech, some medicinal chemists criticized the molecules that they were kind of not diverse, not as novel, not all of the molecules, but one that we took into mice specifically. Many others were actually quite novel. And here we spent about 4 weeks in getting the feedback from medicinal chemists before we decided to prioritize some molecules for synthesis, because at that time China was locked up. And serendipitously, so you would be quite surprised, a lot of my synthetic chemists were in Wuhan. So we order from a very large contract research organization, which does a lot of synthesis. They're kind of the Ferrari of synthesis. They're the best in synthesis. And they're in China as well. So very often you don't know where your synthetic chemists are. I mean, my managers who manage CROs, they know, but I don't. And once we published, we got a very interesting email from Wuhan saying that, oh, Alex, we actually work for Insilico through the contract research organization, and we saw that you published those structures, so we're very thankful for that. And once we get out of lockdown, so at that time they were in lockdown, they couldn't synthesize. They basically said that they're going to be very motivated to synthesize once they can come back. But up until mid-March, we couldn't even synthesize. And the synthetic process is quite long, right? So we are still synthesizing most of the molecules that we decided to prioritize. So out of those 7, only 2 got synthesized, and we have not yet tested those. Well, some are being tested right now in several labs. What we did, we basically used crystal structure, homology modeling to generate the molecules, co-crystallized fragment. So 3 ways of generating. So think of it as we look for a pocket, for a binding pocket on the protein surface, and then we imagine some template molecules in that pocket, and then we expand the chemical space around that template. And then we filter out all the molecules that don't have a chance to work. And then we rank and prioritize. Or we look at the molecule which binds to the protein already. And then we create kind of deep fakes of this molecule with specific properties and make many kind of examples of this, many duplicates with varying properties of that molecule. And also rank and prioritize. So February 5th, we published the website and also submitted to BioArxiv and showed some representative molecules that were actually very diverse. So the next closest molecule to what we imagined using AI in the libraries is only like 80% similar. And right now those molecules are being tested. So that's what we did. And we're still synthesizing them and testing. I think in about 3-4 weeks, we will know what worked in the assays. And afterward, we will need to, so we will get the hits. Those hits that we've generated are very drug-like. So they have much higher probability of becoming good drugs than if we were to find those in high throughput libraries. And then we will have to do animal experiments. And of course, we will need to look at infected culture. So it will still take us, at least a year before we can put anything into humans in the best case scenario. But we're still continuing to do that. So that's what we did on the kind of chemistry side. But also, I realized that the virus is more harmful and infects mostly the elderly people. And put out a paper on gerophilic nature of this virus. And I guess it's the topic for this different discussion. But currently, we are also looking at how to employ AI to generate de novo molecules. We also had a repurposing exercise. So we looked at what already existing molecules that exist for possible efficacy in COVID-19, but we did not publish them yet because we are very afraid that people are going to start using them, right? So this ones you have to synthesize and test. If you put out the unproven, even though they're safe, molecules as a repurposing exercise, a lot of people are going to use those, including doctors. And, we've seen what Trump did with hydroxychloroquine and azithromycin. So a group in Marcel published a study without pretty much any peer review, and that was a weak clinical study. And that study is now one of the most popular papers in the history of human of science, right? So I think the altmetric score is 6,000 to 7,000. So lots of attention, and it became almost a standard of care overnight without substantial evidence. And that kind of deprioritized and refocused people's efforts on those two molecules rather than looking at more alternatives. So it's much more difficult to do clinical trials. So we decided not to do that and first publish this. But then we, of course, so I personally, not as Insilico, decided to publish a commentary and a perspective on the gerolytic and gerophilic nature of this disease.
Very interesting. A lot there to digest. I want the audience to realize that we're going to have the best show notes that we possibly can in the description below. So make sure you check that out to follow along for some links there. Alex, I believe you mentioned in the Aging Journal your article. So that was a fascinating article. I highly encourage the audience to check that out. Looks like you're going to bring that up, but there'll also be a link in the show notes below. There we go. Yeah. And so as you're bringing that up, Alex, can you share with us a little bit about ResTORbio? I know there was a article on lifespan.io about that. Can you share a little bit about that and how that relates to COVID-19?
So sure. Actually, yeah, before I even jump into my paper, it's worthwhile explaining some background on the various geroprotectors that might protect the elderly against the viruses. I must say that before this study, before COVID-19, before SARS-CoV-2 pandemic, I was not so convinced that rapamycin and other rapalogs are so promising in both geroprotection and also protection against infectious diseases in kind of low doses. I liked the molecule. I tried it myself even, and did the blood titrations and looked at how it affects me specifically, just to kind of look at some pharmacological properties of that drug, even though it's very well described. But I was not really convinced that rapamycin is the way to go. But now I am fully convinced that rapamycin and everolimus RAD001 from Novartis and maybe several other rapalogs that were developed by others, also AbbVie, pretty much every big pharma developed a rapamycin analog or derivative. In, I think, early 2000s, people started realizing that rapamycin, which was originally discovered as an antibiotic and then repositioned as antifungal and then repositioned as an immune suppressant for organ transplantation and then repositioned for cancer and then repositioned for many, many other diseases. So it's kind of a magic molecule, not unlike aspirin in the context of, its applicability to many diseases in different doses, different combinations. It's kind of like a Swiss Army knife of your drug portfolio. People started noticing paradoxical effects of rapamycin in protection against infectious diseases. So what I encourage your readers to do is to go on Google. I mean, the best form of AI is actually, Google text search engine, and just search for, let's say, rapamycin, and let's say paradoxical. And you can see that, paradoxical aspect of rapamycin in immunology, there are many, many others. Or Google rapamycin in the context of some other context. So you can see how it works. You can see some anecdotal evidence that it's in addition to prevention of organ rejection, it also potentiates immune response to all kinds of pathogens and all kinds of infections. So in, let's say, influenza.
I've personally been talking about rapamycin for at least 5 years. And yeah, the more longevity enthusiasts that I interview, the more they seem to be, coming around to the benefits of rapamycin, some more than others. But that's kind of the interesting things about a pandemic like this, and how it can totally shift focus of everybody, our global society, governments. Governments weren't prepared for this properly. Many scientists weren't. The Bill Gates TED Talk from 2015 is kind of gone viral now, no pun intended. With him warning about a pandemic coming, and so it's really interesting that were foreseeing this, and how you have shifted Insilico Medicine to be focusing on this. What's going high level real quick? What's the kind of the main things that we should be taking away from how our world has changed so drastically, so quickly here? And it's a really open question. What can you guys learn at Insilico Medicine? What's the message for the audience? And then maybe what's the message for governments and governments that fund scientific institutions like colleges and universities?
So sure, but Brent, before I answer this question, can I fully answer your previous question about the TORC1 inhibition and the failed trial of ResTORbio?
Yeah.
Because I kind of started from very far, and that's one of my side effects, so to speak.
Sure.
I tend to focus on something that might lead to the point, but it takes me a while to make the point.
Well, you have the mind of a scientist, yeah. And so you go into so much detail, and I know the audience, and I appreciate it so much. So yeah, please do your thing.
So failed study by ResTORbio. So let me explain. So first, there was anecdotal evidence of rapamycin's effectiveness in preventing viral infections and also all kinds of infections, despite it being an immunosuppressant. So people noticed that in many cases, it became immunopotentiator, right? And it essentially prevented infection in many patients. And scientists at Emory did a lot of groundwork there in rapamycin, and then also looked at other rapalogs. So highly encourage you to look at Emory's work. And Novartis, which developed a very close analog of rapamycin called everolimus RAD001, and they also took it for organ transplantation and cancer. So tons of clinical studies completed already. So it's a very powerful rapamycin analog. And a lot of people are trying to basically say, no, it's not an analog, it's a different molecule. So actually, let me show you very quickly. Again, I'm kind of trying to make the story, the short story long, but you need to see that, right? Because it's noticeable with naked eye. So let's say we look at sirolimus, and I'll just look at Wikipedia, for example, right? So because it's easy to jump on that. So look at this structure, and let's say everolimus. Let's look at this structure. So it's going to be very visible to the naked eye. And let's say we also do another one. So that's BES 235. I'll tell you what it is exactly. So that's actually, so doctolizib, that's what failed in clinical trials for immune potentiation. So that's what, this is the molecule that ResTORbio took in clinical trials, right? So check this out. This is original rapamycin, original sirolimus. So see the scaffold, right? And kind of look at this group. So very important to look at the scaffold in this group. This is everolimus, that's the Novartis drug. So this is the same. So all you have is kind of minor modification here. So if you're claiming that it's a completely different drug, it's very nice to make this claim, but then you can not criticize, for example, my paper that I published in Nature Biotech, which is actually more different than from its original scaffold, than everolimus is different from rapamycin, right? So just minor modification, right? So they are technically the same, okay?
Yeah.
You can make all kinds of claims, but they are technically the same. It's patentable, but this is the same. But look at this molecule, right? So very different. And it's an allosteric inhibitor of TORC1 complex as well. But it also was originally discovered and developed as a PI3K inhibitor. So if you again look at, it's not to be trusted. I don't like Wikipedia as a source, but here is just for quick reference. So PI3K inhibitor originally, right? And if you look at what Novartis did originally, so they took, so John Malik, again, have immense respect for her, and she presented her original study that she published in Translational Medicine at my conference in Basel, that I co-organized with Martin Scheibe-Knudsen from the University of Copenhagen every year in Basel, called Aging Research for Drug Discovery. So 2014 was my first one, and Jean presented the early work where everolimus alone was used as a vaccine potentiating agent in healthy elderly patients. Then they conducted a follow-on study and showed that a combination of RAD001, so here she combined them and showed in a phase 2A study, randomized placebo-controlled, that there is an improvement in response to influenza vaccination, and it actually improves the immune function in the elderly. Big thing, right? So huge paper, hugely important paper. And then my understanding is primarily because of patent protection. So RAD001 is a Novartis drug. In many areas, it's kind of like going off patent. Best to the 235, it's a much stronger IP position, I think, for ResTORbio. So they rebranded BEST 235 as RTD, so ResTORbio 101, and took that into phase 3 clinical trials for the same thing, for protection against influenza, and failed miserably. So if you again Google lifespan IO, oh, this one. I already Googled it before. So,
it came right up.
Yeah, so that's a beautiful picture, right? Kind of, it's the disappointment for our entire industry, for everybody in longevity. That's a huge huge failure, which starts the rapalog winter in immune potentiation, right? So no investor would touch a rapalog for immune potentiation anymore because of the failure of this. And of course, they can claim that, look, it's safer than sirolimus, it is safer than everolimus, that's why we took it forward. But come on, here in that particular kind of statement, they're saying, look, while those results are disappointing, we're going to try it again in combination with sirolimus. I mean, come on, what's the logic, right? So you're going to combine it with a toxic drug? No, why not just take sirolimus in clinical studies, right? They're going to take it for Alzheimer's, right, or Parkinson's in this case. So come on, it's not, you cannot be just a magician who says, oh, well, sirolimus is toxic, that's why we're going to combine it with RTB 101. No, it's a commercial play, and we understand that, but why not take another derivative maybe? Why not make another derivative of sirolimus, which might have some additional properties that increase bioavailability or specific tissue specificity or something else. So yeah, that's the reason why rapamycin, why ResTORbio failed. I think that they used a different molecule, so they just did not combine it with the everolimus there. And Matt Kaberlein, so one of the scientists I deeply admire, young, wonderful, productive. So if you look at RTB 101 failure, so let's do it like this and see if, this is Mario the scientist, and I don't know how to spell his name. Well, with my name, try that.
Yeah.
So this paper is probably the best in explaining why it failed. So he goes very deep into the subject, and of course talks about how, so rapamycin everolimus, RTB 101, so it basically tells you what this drug is and what it is not. So it doesn't improve vaccine response in people. And rapamycin everolimus should. So it should be tried for protection against COVID-19. So yeah, that's kind of the long answer to your question. Sorry it took so long.
No, and Alex, that was great. You said should. Do you have maybe an age recommendation of where it should start? Because I'm 35. I believe you're right around 40. You said that you've tried it. What's your best recommendation that you can share with the audience about age with rapamycin?
Right. So let's shift gears to my paper first, right? And I published this paper, perspective paper, on geroprotective and cell-remediative strategies to reduce comorbidity, infection rates, severity, and lethality in gerophilic and gerolavic infections. I actually must also make a confession. I did not write this one alone. So, you know, like behind every great study, there is a woman.
Yeah.
And I partnered originally with Rita Radinkovich from Guy's and St. Thomas Hospital. So one very brilliant, prolific young scientist and medical doctor. And she co-wrote it with me, but she couldn't co-publish because she couldn't put her name on this because she's a medical doctor right now at the forefront of this pandemic, right? And at that time, you know, she couldn't even put her name on it, but she contributed to the paper quite a bit. So to some parts, for, you know, describing the existing treatments, she basically wrote them. So for me, it was I'm always now admitting that's the case. So she basically contributed to that and co-wrote it, but couldn't put her name on it. So very, very sad. And if you look at this paper, so we first of all coined a couple terms. So we coined the term gerophilic, more affecting the old people, and also gerolavic. So geros is old man, and epilavis is harmful. So it's harming the old people. And if you look at the data from Wuhan, it was available at that time. Well, we can trust or not trust this data, but at that time we could work with this data. It was the best it was. So if you look at the infection rates in Wuhan, most of the people infected, so 90%, were over 30. And again, most of the people were over 40 and 50. And if you look at severity and lethality, right? So severity and lethality, so 99% of the deaths were over 30. And 95% of the deaths were over 50, which is very correlated with immunosenescence, right? And also the evolution of the thymus. And, you know, if you look at our immune response to infection, so it goes down with age, but not like that. So if you look at influenza, it affects the young and the old in a more fair manner. So this infection is not fair, it hits the old elderly. So which presents us with a huge challenge, but also a huge opportunity for the entire, you know, biogerontology community to go and try geroprotectors to prevent infection in the elderly, and also to treat them with geroprotectors and help them recover more effectively after infection. So this is a golden super case of a gerophilic gerolavic infection, where we should be focusing on the elderly and not on the young people. So when we're talking about, you know, rapalogs or other geroprotectors, I would again call for clinical trials. I wouldn't call for taking them. I would call for a clinical trial to try those for prevention in a very expedited manner in basically 50 plus or 60 plus. So for young people, for you, I don't think you should be taking those molecules currently. Maybe you should for other reasons, but not for SARS-CoV-2. But you can see that 50 onwards, that's where most of the infections are and when most of the deaths are. So that's where you should be looking at potentiating your immune response. And what we also proposed is this kind of schema. So aging leads to immunosenescence, immunosenescence leads to infection. So here you have the chances of death. So you can die from infection. However, this infection leads to more damage, loss of homeostasis. So you're likely to have accelerated aging and accelerated immunosenescence. So kind of vicious circle. Also leads to accelerated aging, age-associated pathologies. If you had any comorbidity, it will get compressed and you are going to die of it or have higher chances of dying of it. So here you also have higher chances of death. So what we really need to go after is immunosenescence. And for people who do not get the disease, you would want to put them on some regimen that potentiates immune response and prevents immunosenescence. But with SARS-CoV-2, we have to be careful because in the early stage and in the acute stage, you've got cytokine storm starting. So that's a very acute immune response, where your own immune system starts attacking your alveolar cells. That kind of leads to fibrosis and those scars that form in lung later on. So it's also important to administer some of those treatments in the right time. So before the disease or after the disease. So I would not recommend even clinical trials for that when the person is in the acute phase. So what I came up with here is various stages of SARS-CoV-2, right? So exposure, so that's where you kind of touch somebody or talk to somebody or got exposed to the virus, we don't know. So it's high risk of infection, or you're living with somebody who is infected, and all the way to kind of recovery phase. So this is kind of the stages of SARS-CoV-2 and then COVID-19. Very important to make the difference, kind of like HIV and the AIDS. So originally, before you get infected, you might want to protect yourself against SARS-CoV-2 infection. And just like with vaccines, you would want to do it early before you get exposed, right? So a vaccine would be administered before you get exposed. When you get exposed or when you actually experience early symptoms, you don't want to vaccine because it could make things worse, right? So same for geroprotectors, you would administer them before you get exposed to the disease. And rapalogs, like again, I would use sirolimus or everolimus. We clearly see that RTB 101 doesn't work, so I wouldn't try that. So let's put that to bed. You would use a known rapalog, very well tested and tried in the clinic, preferably sirolimus or everolimus, in low dose, like 5 to 7 mg once a week, possibly combined with metformin, also with a specific regimen, but that one you have to take regularly over the course of several weeks. Before you get exposed to the disease. So that's when you are trying to protect the elderly and in a maximal way, segregate them and protect them from exposure to the virus. And during that phase, you would want to use those rapalogs and metformin in a clinical study. So I'm not proposing a DIY or even current treatments. It needs to be tried and tested, just like with other geroprotectors, before exposure. And actually, if you look at Sweden, so Sweden decided to protect the elderly by kind of segregating them from everybody else. But life is not as normal, but people are free to operate. And instead of being in lockdown mode, the elderly are being protected. So, you know, countries like that could do a clinical study on geroprotectors to see if it is possible to administer them before infection and see how it goes. And of course, after the virus is the infection subsides, the response to infection subsides, you get into recovery phase and rehabilitation phase. So we know that there is a fibrotic buildup in the lung, even with mild symptoms. So the largest laboratory experiment, human lab, that was kind of involuntarily and serendipitously created the Diamond Princess cruise ship. So we've seen that a lot of people got it. So more than 700 people got the infection, 7 people died at the cruise ship, and a few died later on. So many of those people who experienced mild symptoms, they actually got lung fibrosis. So it's not the lung fibrosis induced by intubation. It's actually the COVID-related fibrosis. So it's important to look at antifibrotics and then look at the rehabilitation, and again, use geroprotectors and again, rapalogs and a few others. But here you can actually use larger arsenal for recovery and rehabilitation. So what I'm also proposing is the use of aging clocks at that time. So once you're potentiating the elderly with a rapalog or metformin or senolytics or some other geroprotectors, you need to start looking at various data types like blood tests, transcriptomes, methylation, microbiome, even pictures, to see how those geroprotectors are kind of shifting the dial on the aging clock in which direction and how much. And that would present us with an opportunity to actually test those geroprotectors as geroprotectors to see if we can rewind the clock or slow it down in a large patient cohort. So even those people, even when those people are never going to get exposed to SARS-CoV-2, we would be able to see if there is any geroprotective effect. And that would be a dream come true. So I also proposed using aging clocks like our very simple aging AI, where you can key in a large number of parameters from your recent blood test and basically test your predicted biological age using those very, very simple blood test parameters that come from your recent blood test and see if before and after, if there is any difference before and after going on a geroprotective regimen. So that's what that's what the paper is all about.
Beautiful. Would the Steve Horvath DNA methylation clock be involved at all in what you just showed there?
So I think it's important to test all the aging clocks.
Right.
So many, and I'll actually change my background to have a more relevant content.
There we go.
So yeah, it's deep aging clocks. We published that paper in Trends in Molecular Medicine. So I highly encourage you to take a look at this paper with Polina Moshina, one of the kind of leading authorities on aging clocks, with the DPhil from Oxford. So that's the picture. And now I can address actually your second question if you like.
Yeah, please do. And we'll try to get a link here to what you have in the show notes. So again, everybody check the show notes here, and you can obviously pause the video to zoom in on that. So yeah, please go ahead, Alex.
So sure, your question was about the economic impact of SARS-CoV-2 and also geroprotectors. So how it's going to change the world. So I'm not going to talk about the obvious things that, more people are going to work from home. There's going to be more attention to, biomedical sciences. And I think that there is a shifting priority from, watching Kardashians to looking at biomedical sciences. So who are the real heroes? I really hope that children who are staying at home with their parents will choose a different career track as well and go into biomedical sciences. And also, I hope there is going to be more attention to aging research, because there is nothing more important than aging research in life, period. So what we are doing currently with Insilico, of course, we're going into drug discovery, but the reason why we're going after that is to create a sustainable business model for aging research, and also to accelerate many processes that we need anyway for aging research. So that's what we're doing. And I highly encourage you to take a look at my book, called Ageless Generation.
Yep, we'll have a link in the description below to the Amazon for everybody.
So in that book, I outline several scenarios actually for human economic development in the context of aging research. So currently, there are many elderly in developed countries, and they actually depress the economy, right? So currently, the more elderly you have, the more healthcare spending you have, and the less productive the country is. And also, if in a democratic world, the elderly are, the very important voting group. So if you can see what was the major similarity between the two presidential candidates last round. So they were both 70, right, or pushing 70. And this round, it's even older, right? So you're not seeing younger people running for president or having a chance. And in that book, I outlined two scenarios. So basically, one where we go as we go without the focus on aging research, and the many biomedical advances are going to extend lifespan, but not the productive lifespan, and the economy can collapse, because right now, most of the developed countries, the economies are not sustainable at all. So there is such huge debt burden in those economies that it's already unsustainable. So if the GDP drops, currently the debt level is so high that nobody in their sane mind should be buying bonds. So we might go into the same scenario as, the Weimar Republic in Germany or Zimbabwe, when there is a period of hyperinflation and there is no trust in the currency, right? So fortunately, right now, the US dollar is the kind of global economic universal currency, and everybody has the trust in that particular currency. But when the printing of this currency goes out of control, and many other countries pursue the same thing, there could be an economic collapse, and that will put even more pressure on the economy, and we won't be able to have the adequate funding going into aging research. I actually published another paper, biomedical progress rates as new parameters for models of economic growth in developed countries, with a brilliant scientist, Maria Litovchenko, currently at EPFL in Lausanne, doing her doctorate. Also under Biogerontology Research Foundation. And in that paper, we show a very, very simple model, very simple idea that imagine there are 3 points in human life: statutory retirement age, in some countries you have that, maximum productive age, and end of life. And those are arbitrary numbers, not something exact, right? And there is a biomedical and technology progress rate that technically shift both maximum productive age and end of life to the right. So in general, we start living longer and more ubiquitously, so in more countries, and the quality of life is increasing, right? So technology is really making our life longer and better. However, if you look at the pharmaceutical drug development nowadays, most of the clinical trials for products out there are focusing on extending end of life. So basically, if the phase, last stage, final stage of prostate cancer, so prostate cancer metastasized to the bone, terrible pain, and we're still looking at ways to marginally increase the lifespan of that person with that metastatic prostate cancer metastasized in the bone by a few months, trying to add additional, 3-4 months to the person's lifespan, while not really extending productive age, right? At that time, you are in terrible pain, walk to a bed. And in the US, I think more than 65% of the bankruptcies are health related. And within, 4 or 5 years of getting cancer, most of the people run out of money. So the current healthcare system is flawed, right? So most of the biomedical advances are focused on this in pharma, versus looking at increasing the maximum productive age. Like, think of an innovation, and it's not only for biotech, it's for everything. Like, if you think about restoring lost function, think about glasses, right? Or LASIK surgery, which restores function in the elderly. So pretty much the same way we could address many other challenges. So what we proposed in this paper is that if your rejuvenation rate, so the rate at which maximum productive age shifts to the right, outpaces the non-rejuvenating biomedical progress rate, the economy should grow at unprecedented rate. And the higher this rate, the higher the rate of economic growth. However, if the non-rejuvenating biomedical progress rate is outpacing the rejuvenation rate, then the economy should be declining, even without SARS. So what we will see likely to happen in the developed countries in the very near future is that the GDPs of the developed countries are going to go down while the debt burden is going to go up. So this economic stimulus, the printing of money, is going to lead to higher inflation and also less trust in the government. And we might see the global economic collapse as the other kind of potential challenge. So that's why I think that, Blaue area in Hainan is the best place to set up kind of a longevity conclave currently, and because just the best place on earth. I surveyed many. And here in this paper, you've got some math behind this kind of new theory of longevity economics, and showing that investments in rejuvenation would yield ultra substantial economic benefits. And kind of coming back to the paper on geroprotection, I think if we had a chance to go after SARS-CoV-2 today and start the clinical trials for geroprotectors and as many as possible, we could have a positive economic side effects that these trials would also show that some of those geroprotective regimen may also shift the productive longevity kind of time point to the right? And we were going to see major economic growth. So coming out of this crisis, we might actually come out stronger if we have proven geroprotective regimens and start looking at, more interesting and more impactful interventions like gene therapy and stem cell and regenerative medicine, and also advance our understanding of aging clocks substantially and understand the value of the various data types to aging clock creation. So I think that's what the world should be focusing on. That's why I put this paper out.
Oh, lots of great stuff in there, Alex. I mean, without being insensitive to how COVID-19 is in fact affecting all of us, and especially people who actually had the disease or loved ones. I mean, the reality is this is a huge opportunity for the longevity movement and scientific community and globe to understand that about 110,000 people every single day are dying from aging or age-related diseases out of about 150,000 people total. So this is the largest problem on the planet if you define a planet as people dying. And I define that as a problem because I'm a person. I don't want to die, and I don't want my loved ones and other super intelligent people to die who are contributing to the global economy. So this is, our chance to really get this message hammered home. And, you just did that so eloquently. And I just really want people to take this as, the number one message to share with the global community, because it's so important. And the work that you and your team are doing is so valuable. And I think, kind of like I said, and when I interview in January, it's just, it's frustrating that so many people are still watching Kardashians or whatever, while, this esoteric, incredible work that you and so many other scientists are doing is kind of just getting overlooked by the mainstream, the mainstream media, even social influencers, social media influencers. So, I really, that's why I want to plug, your social media, follow Alex personally on Facebook, on LinkedIn, on Twitter. He's always posting great information to keep up with the research. We'll have links in the description below. And yeah, I mean, I wasn't expecting you to talk there about, the currency and the world reserve currency being the US currency. Currently, what's been happening in the last couple months since COVID-19 really started having a huge impact is, while yes, inflation is affecting Americans and people who hold US dollars, actually it's inflating less than other currencies. So the US dollar has gone up in value relative to other currencies like the Japanese yen, Australian dollar, the euro, on and on and on. But if inflation is still basically a hidden tax that's affecting all of us. And what's so unfortunate is it's affecting the poor the most. It doesn't really affect, the wealthier people as much. And so, I always try to talk about compassion. The work that you're doing and so many of the scientists that I interview is about compassion. And we need to be thinking about the least fortunate on this planet and what this pandemic is doing to them as well, because that's very important and that shouldn't get lost. And that's also the elderly people, but also just everyday people globally who are struggling financially. So, wow, really good insights from you, Alex, on that. I was not expecting you to start talking about the actual currency and GDP and global stats like that. So thank you for sharing that.
Well, again, I think that, there is Chinese is the most, I would say, insightful and wise language on the planet. And if you think about the word crisis in Chinese, it's here. So it's Wei Ji, and it's comprised of two characters. So this one, so Wei, is, I'm not pronouncing it correctly, I cannot. it's danger, and the other one, G, it's opportunity. So every crisis presents an opportunity, and
Right.
here is the opportunity for the longevity community to really step up. And I think we should start with going after SARS-CoV-2 with a geroprotective regimen, again administered before the exposure to the disease to the elderly. And I think the best way to start that would be rapamycin. So trying rapamycin or everolimus if Novartis is up for it. I would say the best rapamycin formulation currently that I know of is the rapamycin syrup by Pfizer. And if companies like that, so Pfizer could try that for a clinical trial, that would be truly amazing. Of course, it's not as commercially viable as having some very new patented drug that you can later milk for tens of years or for multiple years and make billions of dollars. I would say it should be very similar to targeting aging with metformin study, where we actually combine rapamycin with metformin or just do rapamycin. And again, low dose rapamycin once a week before exposure. Clinical studies should be conducted, and we need to campaign for that. So if Lifespan.io can help facilitate that, we might see an opportunity here.
Definitely. Alex, what's your thoughts on the FDA rapidly approving COVID-19 treatments, or at least much faster than the normal FDA process? And before you answer that, my personal thoughts have been, hey, we already have a crisis. 110,000 people are dying every single day from aging and age-related diseases, and it doesn't have to be this way if we put more research in. This is what I and many people in the longevity community have been screaming. However, at the same time, we want to go through a rigorous process. I just don't think that it has to be as bureaucratic and as expensive and as time consuming. And we're seeing right now, when there is a perceived crisis with COVID-19, that the FDA can, approve things much faster. So the question is, how can we take this environment that we're currently in of COVID-19 and seeing that the government and many government entities and the scientific community in general can work together and rapidly produce incredible results? How can we take that and carry that on forever for longevity research? I know it's a difficult question, but what do you think on that?
Well, for longevity research, to be absolutely honest with you, I don't know. So that would require kind of thinkers like Daria Kalturina and the others who are kind of working hard to classify aging as a kind of disease in context of WHO's ICD-11 and looking for ways to kind of call for clinical trials and many others. I mean, we need to have more people looking at the clinical side of things and trying to call for clinical trials, like Nir Barzilai is doing that for targeting aging with metformin. I mean, we've been talking about that study for over a decade now, I think, or at least, or almost a decade, and it hasn't started yet properly. So we need to take, lessons learned from TAME and try to have more and more studies like TAME in parallel, because we don't have the time to wait now. And lobby the FDA to start those studies. Unfortunately, many of those studies would not be commercially viable. So, large pharmaceutical company wouldn't be able to bank a phase 3 clinical study or a phase 2 clinical study for the asset they don't own that is generic and that could be taken into the clinic by anybody after the clinical study is complete. So we really need government intervention, or we need to have billionaires like Bill and Melinda Gates, Facebook guys, or some other high-level intellectuals with money to bankroll those clinical studies and work with the FDA to develop those studies to complete an accelerated manner. And for that, we need to invest heavily into aging clocks to understand where do those geroprotectors work, on which data type, how substantially those aging clocks can be reversed or slowed down, and collect pretty much every data type. So I think that right now we're seeing the acceleration of clinical studies in the context of SARS-CoV-2, but it's not going to be a sustainable process for many other treatments, and of course not for longevity. So here we just need to take this opportunity to lobby for geroprotector clinical trials, and first let's do the TAME and also try serolimits as soon as possible. So not doing it for a small biotech company like ResTORbio, I think, or some other company focusing on rapalogs, but really taking some generic, very, very well established geroprotector that works in multiple species, like rapamycin, into a clinical study, and trying to accelerate that while we have this pandemic, because while it's ongoing, there is disease out there, so we can see if it really potentiates response. I'm talking about rapamycin, for example, and other combinations. So I think other people, not myself, should be campaigning for that. I'm just showing that there is a sense of urgency and there is an opportunity. And I'm very surprised that there is no outrage from the longevity community for the lack of focus on longevity therapeutics being tested in a clinical study.
Yeah, very well said. I personally am, and you're a scientist and entrepreneur, so you're busy enough working your tail off every day on the work that you're doing. What I'm trying to do, and I know the Lifespan.io and the Leaf team is trying to do, is get this message out to as many people as possible. And so I just want to, plug Leaf and the YouTube channel. I'm really excited to be a volunteer, and this will be a new series where I'll be interviewing, longevity thought leaders in this long format like I've done on my YouTube channel. I really want to ask the audience to please like and share on your social media this interview so you can get this information out to people. And also, it's really important to the YouTube algorithms that you not only subscribe, everybody knows that, but hitting the bell notification and even clicking all, it really triggers the YouTube algorithm so more people can find this information. And as we've discussed throughout this whole interview, there's really never been a more important time to share this information. And this is, an opportunity for the longevity community to share this and how the kind of research that Insilico Medicine and Dr. Alex Zhavoronkov and his team, and so many other folks who have tirelessly been working for years and decades looking for more funding, that they really do deserve and can make a huge impact. This is the opportunity for the world to hear this message. So please do that. It's a really easy action item on top of the likes. And please let us know in the comments below, what you guys thought about this interview. All constructive criticism, feedback is welcomed, and we'll take that into consideration moving forward. Alex, what other final thoughts do you want to share with us? Because you've been we appreciate your time, and your time is very valuable. So, what are the final thoughts about COVID-19? What you're working on? Any kind of final thoughts that you want to leave with the audience about what we discussed?
So sure, I think that now that a substantial part of the human population in developed countries is in lockdown mode, it presents an opportunity for the many to study longevity, right? To understand the longevity industry, to understand the landscape, to contribute to projects like Lifespan.io and many other nonprofits. There are a huge number. So I think people should find ways to volunteer for those projects to actually deeply understand what's going on. They might not immediately form an opinion on where to go, and it will take time. So I'm in this field for over 17 years now, and I'm still learning every day and changing opinions every day as new data becomes available. Again, very nice shift into rapalogs now.
Alex, on that note, real quick, in the last 3 months since COVID-19, if you had to say, what's your biggest opinion that you've kind of changed? It could be anything, not just how you're kind of viewing the whole business model of Insilico Medicine. Maybe how is that kind of changed?
Well, for Insilico Medicine, the business model is not going to change dramatically, because we really need to deliver on our promise to the investors. And as a CEO of a company, you have a fiduciary duty to deliver on the promise and on the strategy. So, of course, we pivoted into SARS-CoV-2 as well. However, for us, business is as usual. So we're progressing the preclinical work in multiple areas, primarily fibrosis and cancer, and also one antibiotic. So antibiotic focusing on microbial diseases. We have 9 programs currently. All of them are progressing. We are progressing in AI. However, we also decided to spin off our aging clock business into a separate company. So I hope that's going to be big news coming up in the future. Doing a lot of work there. So a small part of the team is going to go into aging clock business, and also this team has a lot on their plate in SARS-CoV-2 now. So my kind of biggest change in opinion was in rapamycin. So I started digging very deep into rapalogs, and I did not really understand the reasons for failure of ResTORbio. Now I do. So thanks to Matt Kaberlein again. So sorry for misspelling his name.
K8. We'll all remember now.
Great guy and great scientist. So everybody should read that paper. And I think we need to start clinical studies on rapalogs. I think that we need to invest more heavily into deep aging clocks and start going to there. And again, my advice to everybody on the call who are still listening, which is unlikely, but if you're still here after this lengthy video interview, if you're still there, just join one of the volunteer organizations, or actually become a Wikipedia editor. That's a great thing. I think that this industry lacks Wikipedia editors. I must say that one of the editors there, his name I think is David Gerard, at some point of time got into quarrel with me and kind of started engaging his entire kind of army of friends to remove the content on Insilico and on me personally. So I just decided to even not look at that anymore. That was pretty frustrating.
Yeah.
So if you guys are working as Wikipedia editors, volunteers, just edit some aging sites, get into rapalogs, look into Synalytics, look at NAD boosters, look at metformin, look at other ways for SARS-CoV-2 prophylaxis using geroprotectors, call for clinical trials, start learning the various fields of biomedicine by reading papers, also again editing, putting blogs out, editing Wikipedia, getting the right knowledge, and ensuring that the facts are right. Don't promote hydroxychloroquine and azithromycin or trials like that or drugs like that without proper clinical trials. I think everything should be tried in a proper clinical trial. Take a data science course, and get more expert in longevity biotechnology, because you can be the most important person on the planet if you contribute to aging research in the most impactful way. And we don't know what the most impactful way is. It's the most important topic. That's what should be driving you, not Kardashians, not a movie, not a video game, not marriage, not kids, longevity.
Beautiful. Very empowering and very impactful. So, Alex, we really appreciate your time. We hope that we can have you back on lifespan.io and Leaf's YouTube channel again. Maybe you can share an update with us as COVID-19 pandemic situation unravels, and maybe when it's more normal, in this world, we can focus back more on Insilico Medicine, and you can share what the team has been up to, because I know it's going to be a lot of exciting updates in the months and years to come.
Great, and thank you very much for volunteering to do this kind of stuff, because again, like Leaf, like your audience, like you, and let's continue fighting with aging, because everything else is a waste of time.
Yeah, I agree. All right, well, thank you so much, Alex.
Thanks.